Hello students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is the third section of the chapter Excretion and Osmoregulation, that is excretory products and their elimination. In this section of presentation, we'll be studying about the kidney histology. Okay, so let's proceed towards our topic, that is kidney histology. Okay, we'll be studying about the structure of kidney today. Okay, as you can see that kidney is reddish brown in nature. It is bean shaped, just like rajma you can say, and it is enclosed in a tough 3-layered fibrous capsule. Okay, it is situated between the level of the last thoracic and the third lumbar vertebra. Length of a typical kidney is about 10 to 12 centimeter. Okay, and width is about 5 to 7 centimeter and thickness is about 2 to 3 centimeter. And the average weight of typical kidney is around 120 to 170 gram. Okay, the kidney is covered externally by fibrous connective tissue, which we call it as renal fascia, and fatty adipose tissues. Okay, if we see the LS, this is the LS section of kidney that shows two distinct regions. First is cortex and inner one is known as medulla. The cortex is dark reddish brown in nature. This is dark reddish brown in nature and granular region. The medulla is lighter and striated region. Okay, medulla, it is not visible in this, but yeah, medulla is somewhere here. Okay, and cortex is the outer one. So the medulla, it shows about 8 to 15 conical projections, which we call it as renal pyramids, and we also call it as renal column of Bertini. The kidney shows more than 1 million microscopic convoluted tubules, which we call it as nephrons. Nephron is a structural and basic unit of kidney. Nephron is also known as uriniferous tubules. They are surrounded by dense network of capillaries. And a typical kidney has inner notch-like structure, which we call it as hilum, from where renal artery enters and renal vein it moves out, and from there renal pelvis comes out, and which extends and forms ureter. Okay, now comes structure of kidney. As I have told you, see, this is medulla, this is the medulla part. Okay, and this has 8 to 15, 1, 2, 3, 4, 5, 6, 7. In this diagram it is shown only 7, but it is around 8 to 15 conical projections, which we call it as renal pyramids. See, this is pyramid like structure, so that's the reason we call it as renal pyramids. The extension of cortex into medulla is called as renal column of Bertini. Okay, and each pyramid, see, each pyramid, this pyramid I'm talking about, each pyramid has a broad base. See, this has broad base that is attached to the cortex and the narrow apex, which we call it as papillae. Okay, papillae. The pyramids show striation which pass radially towards the apex. See, this is the apex and this is the broad base. So, and these are the line-like structures are called as striations. So each pyramid show striation which pass readily towards the apex. The pyramids open into a common cavity called as renal pelvis. See, this is renal pelvis. This is a common cavity in which all the pyramids, they open up into renal pelvis. The outer border of the pelvis is divided into open-ended pouches, which we call it as major calyces, and which further divide into minor calyces. Okay, these are the calyxes. Okay, and these calyces, they collect urine from each pyramid. They collect urine from each of the pyramids, and it collect urine here. Okay, and the pelvis opens into a ureter through hilus. This is the inner notch, which we call it as hilus, as I have shown you in the previous diagram. Okay, so the kidney, it shows more than 1 million microscopic tubules, which we call it as nephrons, and they are surrounded by denser network of capillaries. Okay, hilum leads to a funnel shaped cavity. This is the hilum part. Inner notch is called as hilum. Hilum, it leads into a funnel shaped cavity, which we call it as renal pelvis. This is the renal pelvis with projections called calyxes, and then it moves into the ureter. See, this is the ureter. Okay, I have told you that each kidney has two parts. First is outer one is called as cortex, and the inner one is called as medulla. Medulla has few conical projections, which we call it as renal pyramids. Renal pyramids is also known as medullary pyramids. See, I have shown in this diagram that they are medullary pyramids. Okay, and they project into the calyces. The cortex is the outer part of the kidney. Okay, so the cortex, it extends in between the medullary pyramids as renal columns, which we call it as column of Bertini. Okay, now comes, let's see structure of kidney at a glance. That it has two parts. The outer one is called as cortex, and the inner one is called as medulla. Medulla consists of 8 to 15 pyramid, which we also call it as medulla. Okay, and they open into a calyxes. Okay, and these are the minor calyxes, and they collect urine here, and then it moves into the, finally it moves into the ureter. Okay, and this is how it takes place, that from a renal artery, segmented artery like this, it happens to the renal vein. Okay, so this is how it takes place. Put up that how blood is filtered. The filtering units of the kidney are called as nephrons. They are approximately 1 million nephrons in each kidney. The nephrons, they are located within the cortex and the medulla of each kidney. And the tube of the nephrons, they are surrounded by cells and a network of blood vessels. They spread throughout the tissue. Therefore, material that leaves the nephron enters the surrounding cell, and it returns to the bloodstream by a network of vessels. Now, what are the parts of nephron? Each nephron consists of following parts, which are those: glomerulus, Bowman's capsule, proximal tubule, loop of Henle, distal tubule, and collecting duct. I'm once again repeating that nephron is a structural and the functional unit of the kidney. It is about 40 to 60 nanometer in length. Structurally, it consists of two main parts, which we call it as Malpighian body and the tubular region. Malpighian body is also known as renal corpuscles, and it is made up of two components such as Bowman capsule and glomerulus. Okay, and the tubular region of the kidney consists of following parts. First is proximal tubule, second is loop of Henle, third is distal convoluted tubule, and fourth is your collecting duct or terminal duct. Terminal duct and collecting duct, they both are same. So don't get confused with this collecting duct and the terminal tubule. Okay. So I'm again repeating that each nephron is divided into two parts. First is made up of two parts, glomerulus and Bowman's capsule, and another second part is made up of these four parts: proximal tubule, loop of Henle, distal tubule, and collecting duct. Okay, now we'll see what happens in Bowman's capsule and how it gets blood gets filtered. So items that filtered out of the blood into the Bowman's capsule are four. First is glucose, second is urea, third is salt, and fourth is water. Glucose, it must be returned to the blood by active transport at the loop of Henle. But this is something that is filtered out of the blood into the Bowman's capsule. And second is urea, salt, and water. Okay, now we'll talk about the items that remain in blood. Okay, in Bowman's capsule is RBC, WBC, platelets, larger chemicals like hormones. Now we'll be studying about the part of the nephron and function 1, that is in Bowman's capsule. Force filtration in Bowman's capsule. It causes much of the water and the ions and the smaller molecules to leave the blood, and it enters the proximal convoluted tubule. Okay, see what happens in Bowman's capsule, as I have shown you in the diagram, that Bowman's capsule is a cup-shaped structure, and it is double-walled structure. Its outer wall is called as parietal epithelium, and it is made up of flat squamous epithelium. The inner wall is called as visceral epithelium, and it is made up of specialized cells, which we call it as podocytes. Podocytes, they are modified squamous epithelial cells. They produce foot-like projections, which we call it as pedicles, which extend up to the glomerulus. Okay, and glomerulus is what? It is a knot of the blood capillaries present in the cup of the Bowman's capsule. Okay, I have shown you the diagram also. And then as I have told you that Bowman capsule, it forces filtration in the Bowman's capsule. It causes much of the water and the ions and the smaller molecules to leave the blood and enters the proximal convoluted tubule. Now what happens in proximal convoluted tubule? I have shown you that peritubular capillaries, they contain blood in a hyperosmotic state. So much of the water filtrate re-enters the blood by osmosis, and that is the second step of the mechanism of urine formation, that is reabsorption. Active transport also returns sodium chloride following passively. Okay. Now comes loop of Henle and distal tubule. What happens in loop of Henle? The ascending loop actively transports chloride ions into surrounding area, and thus it recycles salt and creates a hyperosmotic state in the kidney medulla. And the hypertonic state is further increased by urea, which diffuses out of the collecting duct. So you can say that loop of Henle is a middle U-shaped tubule that is situated in the medulla. It has two limbs, such as descending limb and the ascending limb. The descending limb has initial part thick and broad with columnar and cuboidal epithelium, and later part is thin and narrow, lined by squamous epithelium. Okay. And please note one thing, that a desert and mammal such as camel have a very long loop of Henle. Why it has long loop of Henle? That's because to conserve water. Okay, because there's a scarcity of water in desert areas, and that's the reason desert mammals have a very long loop of Henle to conserve water. Now comes distal tubule. Distal tubule is a coiled tubule which is present in the cortex, and it is lined by cuboidal epithelium. The microvilli are absent in DCT. DCT is distal convoluted tubule, okay? And it is lined by cuboidal epithelium, and DCT is close to glomerulus of the same nephron, and it forms justa glomerular apparatus. Now comes collecting duct. In this, what happens is water leaves the collecting duct in response to antidiuretic hormone that we call it as ADH, which is secreted by posterior pituitary in response to osmotic condition in the blood, actually detected by hypothalamus. Now we'll see and it will be more clear to all of you that nephron, each kidney has nearly 1 million tubular nephrons. Nephrons are a structural and functional unit of kidney, and it has two main parts. First is glomerulus and another is renal tubule, okay? First, we'll study about the glomerulus here. What is glomerulus? I have told you that glomerulus is a knot of blood capillaries. This is glomerulus, okay? And what is this? This is the Bowman's capsule. So we are studying about the glomerulus. So it is a tuft of capillaries formed by afferent arterioles, and the blood from the glomerulus is carried away by efferent arterioles, and this is afferent. From this it enters and forms a tuft like capillaries, and from efferent arterioles the blood is carried away by an efferent arteriole. You can say that glomerulus is a knot of blood capillaries which is present in the cup of Bowman's capsule, and the renal artery that enters the kidney, it divides to form branches which we call it as afferent arterioles. This is afferent arteriole, and each afferent renal arterioles, they enter the cup of Bowman's capsule, and it divides and redivides to form a knot-like structure of about 50 capillaries, which we call it as glomerulus. The blood, it enters the glomerulus through afferent renal arteriole. It leaves the glomerulus through efferent renal arteriole. The diameter of, as you can see, the diameter of afferent arteriole is larger than that of the efferent arteriole, because the efferent arteriole, it then forms a network of capillaries around the tubular region, and these capillaries are called as peritubular capillaries. See, these efferent arteriole will go here, and it will form a peritubular capillaries, okay? Now comes renal tubule. Renal tubule begins with the double wall cup like Bowman's capsule, which encloses the glomerulus, and so you can say that renal corpuscle. Renal corpuscle is what? It is glomerulus plus Bowman's capsule. I have written here wrong. It should be it should be renal corpuscle and not the renal tubule. Okay. So glomerulus and Bowman's capsule together, it forms a Malpighian body, or it is also called as renal corpuscle. Okay. Don't get confused with both the names. Now comes the tubule, renal tubule part. Renal tubule part is the second part of a kidney, and it is made up of following parts. First is proximal convoluted tubule, and then a loop of Henle, and then distal convoluted tubule, and finally collecting duct also comes into it. Okay, now let's see this diagram once again. I have told you that kidney, or you can say nephron, is divided into two parts. First is Malpighian body and second is renal tubule. So this is renal tubule and this is the Malpighian body. Malpighian body is what? It is glomerulus plus Bowman's capsule. And what is this? This is renal tubule. And renal tubule has different parts such as proximal tubule, loop of Henle. This is descending loop of Henle and this is ascending loop of Henle, and it is DCT, and this is collecting duct finally. And collecting duct is also known as terminal duct. Okay, before that I would like to tell you that this loop of Henle is like a hairpin like structure. Okay, it is a hairpin like structure. And then comes distal convoluted tubule, that is DCT. This is distal convoluted tubule. Of many nephrons, they open into the collecting duct. Then this is collecting duct. So collecting duct, it extends from cortex to the inner part of the medulla, and they converge and open into the renal pelvis through medullary pyramids in the calyces. Okay, as I have shown you in the previous diagram, previous diagram. Now it is clear that Malpighian body, which is the first part of the nephron, and then comes the second part, that is renal tubule, which is made up of PCT, DCT and collecting duct. So Malpighian body, PCT and DCT, they are situated in the renal cortex region, and loop of Henle, they dips into the medulla region. I have told you that kidney has two main parts, cortex and medulla. So the Malpighian body, PCT and DCT, they are situated in renal cortex region, whereas the loop of Henle, it dips into the medulla. And the efferent arterioles emerging from glomerulus, it forms a fine capillary network, which we call it as peritubular capillaries around the renal tubule. Okay, and there is one more vessel is found. It is a very minute vessel that run parallel to the loop of Henle. This is loop of Henle, and it runs parallel to the loop of Henle, which we call it as vasa recta. Okay, now comes type of nephron. It is seen that two types of nephrons are found, which are those? First is cortical nephron, and second is juxtamedullary nephron. Okay, cortical nephron, in majority, if we see in majority of nephron, the loop of Henle is too short and it extends only very little into the medulla, and such nephrons are called as cortical nephrons. That means the loop of Henle, as we just saw in the previous diagram, that the loop of Henle, it dips into the medullary part, but in cortical nephron, which is a type of nephron, loop of Henle is too short and it extends only very little into the medulla. Such nephrons are called as cortical nephrons. Now comes juxtamedullary nephron. In this remaining 30% nephrons, the loop of Henle is very long and it runs deep into the medulla, and that's the reason they are called as juxtamedullary nephrons. Okay, and vasa recta is also present in it. Now the question arises, why doesn't water on outside go back in? The water that left the descending loop of Henle, I have shown you the two loops, ascending and the descending loop of Henle. So the water that left the descending loop cannot enter the ascending loop because it is impermeable to water. As the water concentration in the filtrate decreases, the chlorine concentration increases and it causes it to diffuse out of the tubule. And this process is helped by the electrical attraction of chlorine to sodium. Now the filtrate, it moves into the distal tubule where tubular secretion takes place. Tubular secretion is the third step of the urine formation. Okay. Now active transport is used to pull hydrogen ions, creatinine, drugs such as penicillin out of the blood and into the filtrate. Fluid from a number of nephron, it moves from a distal tubules to the common collecting duct, which carries what now be called urine to the renal pelvis. Now please note that since the kidneys, they control what leaves and what remains in the nephron, they maintain the level of water, iron and other materials nearly constant, and that's the reason that it maintains homeostasis condition in the human body. Okay. Now how does kidney does its job? It is the summarization here. Okay. Kidney action is, what happens is blood enters Bowman's capsule through tiny artery, which we call it as renal artery, and then the renal artery, it splits and forms a tuft or the knot of capillaries, which we call it as glomerulus. So the blood pressure, it forces some blood plasma and a small material into the surrounding capsule, which we call it, this is called as nephric filtrate. Large particles such as blood cells and protein, they remain in capillaries. The nephric filtrate is pushed out of the capsule and into the proximal tubule. This is where reabsorption takes place. Reabsorption is the second step of the urine formation. Okay, and then only materials needed by the body are returned to the bloodstream. For example, 99% of the water, all glucose and amino acids and many salts are reabsorbed by the blood, you can say, and osmosis, diffusion and active transport, it draws water, glucose, amino acids and ions from filtrate into the surrounding cells. Some villi-like projections are also there, which help in increasing the surface area, and it also helps in the active transport of glucose out of the filtrate, and it speeds up the reabsorption process. And from here, these components return to the blood stream. Okay. Now the filtrate, it reaches the end of the proximal tubule and the fluid is isotonic now, and the glucose and amino acids have been removed from the filtrate. The filtrate then moves to the loop of Henle, whose primary function is to remove water from the filtrate by osmosis. And then there is an increase in sodium concentration in the cell of the loop of Henle as we move from the area of the cortex to the inner pelvis of the kidney, and this causes water to be drawn from the filtrate. Now comes the function of kidney. The principal function of kidney is to filter blood in order to remove cellular waste products from the body. Okay, and what is the second function? At any given time, 20% of blood is in kidneys, and human can function with only with one kidney also. Okay, and if one ceases to work, that means if one kidney ceases to work, the other increases in size and it handles the workload. Okay, and it can easily handles the workload. Now kidney has other functions also, but it is usually associated with this excretion of cellular waste such as urea, uric acid, creatinine. Urea, what is urea? Urea is a nitrogenous waste which is produced in the liver from the breakdown of protein, and it is the main component of urine. And what is uric acid? Uric acid, it is usually produced from breakdown of DNA or RNA. That means when DNA and RNA it breaks down, then it forms uric acid. Okay, and then creatinine. Creatinine is a waste product of the muscle action. So these are the three things that kidney is associated with the excretion of cellular waste, such as urea, uric acid, and creatinine. All of these compounds have nitrogen as a major component. As I have shown you, then urea, even urea has nitrogen in it. Even creatinine has nitrogen in it. Even uric acid has nitrogen in it. So the kidney, all these compounds have nitrogen as a major component. So the kidney, they are more than excretory organs. They are one of the major homeostatic organ of the body. They control water, pH, and they secrete erythropoietin, that is a hormone that stimulates RBC production, and it activates vitamin D production in the skin also. Okay, so kidneys, they control water, they control pH, they secrete erythropoietin hormone, and it activates vitamin D production also in the skin. And now you can see that kidneys are more than excretory organs, because they can maintain homeostasis also in the human body, and they control water, pH, and it can secrete hormones such as erythropoietin, and it can activate vitamin D also production in the skin. So now it is clear that kidneys are more than excretory organ, and that is why a doctor can tell so much from a urine sample. So this was all about the kidney histology. Thank you, and keep watching Edupedia Word videos.